to the session on the challenges of tomorrow's medicine. The latecomers come in first. So what are the challenges of tomorrow's medicine? When I teach my students, I often ask them if they know what was life expectancy in the United States in 1900. And it was 49 years. Since then, life expectancy is considerably longer, but there is contributions to that, not only of medicine, but also of our way of life, prevention, and technology. But how will medical clinics look like in the future? How will tomorrow's hospitals be managed? To what extent will technology breakthroughs in gene research and nanotechnology and stem cells and artificial intelligence affect tomorrow's medicine? We are privileged to have with us today several experts in different aspects of these challenges and the changes that are expected to come as a result of them. And I'd like to start by looking at the very sad aspect of medicine, which involves neurodegenerative disease. In many aspects, we are today with neurodegenerative disease where cancer was 50 years ago. That is, diagnosis is far too late, people are shy to talk about it, and because of that, when treatment starts, it can only be palliative, it can only help people survive as sick individuals, but not really cure. So what is needed is new, earlier diagnosis methods so that newer and better technologies for therapy will be developed. And the problem is that human beings are different. So to find out what are the underlying mechanisms, what causes disease, you need a lot of individuals, which is a difficulty. So what we thought in our university is to try to find an alternative way to look for alternative medicine. And I'd like to tell you about one experiment that we have done together with Chagai Bergman, who's a professor of neuroscience at our university and brain center, and Dr. Tzvi Israel, who's a neurosurgeon specializing in Parkinson's disease. And I need to find out how this works. Okay, fine. So what is Parkinson's disease? It kills nerve cells in one particular small region of the brain and impairs movement and causes real difficulties to about 1% of the population above the age of 65. There is a treatment that is done in the Hadassah Medical Center, which involves implanting an electrode, just like a pacemaker. This is a brain maker. The battery is put over here, and the patients stop tremoring. And we thought that the main problem with sick individuals is that we cannot test them before and after they got sick, because we only know that they got sick after. But these individuals are treated so that we could test the same person before and after the surgery that improved their condition. And it does marvels in improving their conditions. And the next thing we thought is that because this is a disease that affects all body functions, we could see differences in the patterns of genes that are expressed also in the blood cells of these patients. And that new technologies, genome technologies, little chips where we put DNA and we can find out the total profile of gene expression, could be informative in that aspect. And the patients do really much better. This picture is of an engineer in the Hadassah Medical Center who got sick at 58 when he was treated. He just came back to work after he really couldn't move. So here is our experimental design. We take little glass chips with millions of points of information on each, and we analyze Parkinson patients before and after deep brain stimulation surgery, and we even asked some of them to disconnect the battery just for an hour, and then we can identify the effects of the stimulus many months after the surgery. Of course, we need healthy controls. We often use professors because there are many of them, so that's no problem. Yeah, absolutely. And we were very careful to make sure that there's matching the age, the weight, everything. We separate nucleic acids from blood cells from these patients. We put them on these glass chips, and we designed a signature. We found 29 genes that are expressed differently in healthy versus controls, before and after surgery, before and after disconnection of the battery. And then we asked the computer, can you segregate the samples to different groups? And to our delight, the computer separated the sick individuals together with those with disconnected battery, and the healthy individuals to those with those that went through the operation. Finally, the extent of change that we see in these particular signature genes reflect the efficacy of the neurological success of the surgery. Okay, so suppose we have a reflection of neurological efficacy of this treatment. What would be the next challenge? And the next challenge would be to find out which genes were changed. There is a new and very exciting family of tiny genes. That orange structure that you see there is a tiny gene, 22 building blocks only, compared to an average of thousands for normal genes. And they block other genes from being expressed by wrapping around them. So what we've done was to find out those microRNA genes that are modified in blood samples from patients. Then the computer can tell us which are their target genes, how do they interact, in which domain in the sequence, and whether the change is up or down. So we now have a network. The only challenge that we have now, which is a great challenge, is to find out which of these genes is the one button on which we can press to stop the effects of the disease, just the way the deep brain stimulation did. So we have at least one gene from those in the list that is already known as involved with Parkinsonism. And this is a PhD project of one PhD student looking at the molecular mechanism together with the clinical team and the researchers team, looking for hierarchical pathway. And by the way, today is the day of my students to show their studies in the booth at the presenters hall. So anyone who's interested, you're very welcome. Thank you. Now it is my pleasure to present our next speaker, Dr. David Agus, that some of you might have heard in the panel just before us. Dr. Agus is the director at the University of Southern California Center for Applied Molecular Medicine, and he specializes in cancer medicine, and he's a professor of medicine and engineering in the University of California, and a co-founder of relevant companies developing cancer therapeutics. Dr. Agus.
Thank you. Thank you very much. January of this year, I published a book, and January 15th, the first day the book came out, I went on a television show in the United States called Nightline. We did 3 consecutive Nightline episodes. The first, the anchor, Bill Weir, came into my office. What? I can't hear you. Yes. I don't know if we can make the microphone work. All right. We'll use this. So Bill Weir came into my office, and live on TV, we sequenced his DNA. And to sequence his DNA, to look for the relevant genes associated with disease, costs about $25 to $30. We did that and showed that he was high risk for heart disease. So based on that, live on TV, he's a 42-year-old healthy guy, 3 young kids. We put him in a scanner and looked at his heart. And we showed that he had a blockage of 80% in the big artery going into his heart. And so we identified heart disease early. He would have had a heart attack, whether it be 1, 2, 5 years out. It identified it, and we stopped this disease, and we're able to make changes in him to hopefully delay. I apologize. To hopefully delay that from ever happening. The next morning, the American Heart Association issued a press release. Agus screwed up. And they said, Bill Weir wasn't eligible for screening. He didn't meet our criteria, so why did you do it? I said, are you serious? He was positive, so your criteria are by definition wrong. And they said if everybody was screened, there'd be chaos. Realize that a heart scan is $79 in the United States. And if that really is the case, the American Heart Association should be lobbying for better, scalable technologies with less side effects, etc., instead of saying, don't try to save someone's life. So genetics are one piece of modern medicine. And so if you're in front of a Chinese restaurant, 2 of them, and you look at the ingredient list in those restaurants, it's exactly the same. You taste the food, they can be dramatically different. So genetics are your ingredient list. What the food tastes like is what we call proteomics. It's looking at the proteins in blood. Proteomics is a very important field in today's medicine and also going forward. Realize that in 1976, if you or your spouse thought they were pregnant, we took a tube of blood from her and we went to a hospital, because it could only be done in a hospital, and we injected it into a rabbit. 5 days later, we sacrificed the rabbit. If the rabbit's ovaries were enlarged, she was pregnant. Well, that was the state of the art in 1976. 1977, along comes the first proteomic test. Well, rabbits of the world rejoiced. And at the same time, we dramatically changed medicine. Because women now knew early on they were pregnant. They could take appropriate behavior modification, smoking, drinking, take the right nutrients, diet, etc. Kids, early childbirth, we were able to better care for these kids, dramatically changed medicine from one proteomic test. Well, we now have the ability to look at the entire system. Right? You've got inputs in you. It's your DNA, it's where you eat, it's your spouse, it's where you live, it's how much you exercise, and the output is how you feel. Well, that middle, we call it state function, has been historically hidden. And for the first time ever, We can take a drop of blood and we can look at that state function. And that's proteomics. Well, the next technology is wild. So get a load of this. Each of you have 10-fold more bacteria in your body than you do cells in the body. These bacteria, they control your metabolism, your hormone level, they control how you metabolize your food, many things about you. Yet, for the first time in the last 2 months, we've actually characterized them. First time ever. And we're starting to see that there are thousands of them in each of us. And they're modulatable, they're changeable. So much so that if someone in China, you look at their risk factor for breast and prostate cancer, it's 1/10 of people in Europe or the United States. After someone from China moves to one of those places, after a decade, it starts to approach ours. We always said it's the fast food and all of that. No, it's the microbiome. They adapt to our microbiome and their hormones levels change. We know that now. In fact, breast cancer in the United States goes by latitude because microbiome, the bacteria, change by latitude. And going forward, we're going to actually be able to modulate these things, change these things, and that's going to be a big part of medicine going forward. So each of these technologies is a dimensionality. There's DNA, there's protein, there's the new concept of metabolites that we're going to start to be able to measure in the blood. There are all of these changes that we can start to do it. In the physics world, they call something coarse graining, where there's so many things upstream, they take one downstream element to reflect all of those things. We have to start doing that in medicine. So my hope and my dream is that medicine going forward is much more like climate modeling, where that doctor can be like a climate modeler. Instead of looking at every little value, they look at the shape of the clouds, the wind speed, and the temperature, and they can predict the weather. I hope that happens in our field. So thank you.
And our next speaker represents here the United States government. The Department of Defense runs today studies towards protecting not only soldiers but also the general audience, citizens. And we are really privileged to have Professor Giora Feuerstein here, who's the Chief Medical and Technical Officer in the United States Government Department of Defense Research Unit, to tackle these issues. You okay?
Take the computer first.
Yeah, okay.
Good morning, everybody. It's a pleasure to be here. I'd like to thank to Professor Sorek for the invitation and the organizers too. So the title of the symposium is, of course, Future of Medicine. But we are in the city of Jerusalem and not Delphi in Greece. So we're really not going to predict a lot, but we want to give you a sense of what's important to do, what may have started to do, and especially what the capability to underwrite the future might be. So I'll talk about 3 domains. By no means all that is going to happen in this century and beyond. It's 3 domains that are very important to impact on the success of improving medical health and treatment. So let's get to... So, for point number 1 is our medicine, where they are coming from, and how good they are, and how they move in order to address the unmet medical need and give us much better, potent, specific, and safe medicines. So, I would say in the matter of where the industry is right now, you can see on the left upper side a simple graph that shows in the insert that the productivity of the industry for new medical, biological, and small molecule is actually stagnating in the last decade. The productivity continues. There are some new medicines that are coming up, but it's about the same. The important event is that when you look in the blue bars, it's very clear that the expenses to realize those productivity levels at the same level are exponential. That means that the business model of the pharmaceutical private industry to produce medicine is non-sustainable. And the response to that situation is very clear. The industry responds, and responds all the time. It's a little bit dramatic, but that's what they do. They buy each other out, and the consequences, the number come down, professionals are being laid off, and they cannot meet the unmet medical need. And in fact, in all major chronic diseases, there's no progress for about 2 decades right now. So population ages, the problem exacerbates, and the gap between the needs and what is being produced is widening significantly. So we have to acknowledge that the pharma, private, those who are responsible to produce new drugs, don't meet the expectation. So we need a new business model. And the industry, some of it started to shape up, and to many of you in the community here that are coming from different organizations than the private industry, the offering is here that the only model that can replace the stagnation and loss of capability is a consortium model where the government has to step in, especially governments that hold national health services. They have to provide databases and large population capability for analysis. They also have to produce globalization of medical health information, anonymized, but globality. That will solve the rare diseases, orphan diseases, because if we have a case of 1 to 100,000 or 1 to million on a global scale, it's a very good situation to produce medicine. So governments, national health services have to be part of the solutions that we seek. Academia has to step in. Academia is an entity that has reductionist thinking, mechanistic thinking. They bring the innovation, the new molecular target, and mining the human genome. So broadly, we need that community to join. And of course, the private industry, and as the table shows to you, every entity from that consortium brings a strength. These are the red pluses that is unsurpassed by anybody else. If you put a sigma to those capabilities, you create a model that probably can solve and actually leap forward and close the gaps that is mounting decade after decade. So that's one important thing that must happen in this century. It started to happen by some, but don't mean it's complete. So remember, your government has to step in. Your academician has to get out from the ivory tower and step in and not leave the private industry to show up a burden that they apparently cannot. The next slide is coming to the second topic, which is we move away from the medicine as it is practiced today. When you have a problem, you go to the doctor, and the doctor makes his best, we believe, to understand what your problem is and give you some remedy, if he can. But that has to change because it does not basically address the causes of diseases. So we have to move for predictive and preventive. So the slide shows you if you want to prevent something, you have to know what's coming at you. We have today capability to know that. So in the 4 upper boxes, the world doesn't care. People talk about gene and proteomics, things like that. We have the capability to know from birth, before the birth actually, we can know what this embryo may look like in the passage of life. In order to be able to deal with the aging problem, we have to start at the embryonal level. We can get cells, we can get material, we would know what the makeup. Certainly the blueprint at that stage. When a baby is born, okay, we do not discard his placenta and his umbilical cord, we collect all of it. We collect the person's own cells and we create his bank of replacement treatment, engineer cells, organ engineering and so forth, in order to get that passage right and be able to step in months and years and decades ahead of a problem like Alzheimer and other things. We would be able to know about that risk by looking into various components that make it up in early stages of life. So for the first book, I would say integrated system and following all the makeup of the embryo, the newborn, as people get to the adulthood and then to aging. This package of information needs to be always on board, and as my previous speaker has indicated, the capabilities are all there. And what would realize it is basically the law said, we will be able to repair, we will be able to replace, we'll be able to integrate the system. So this century is about moving away from you have a problem, you complain, you go to a doctor, and maybe he may be able to do something. Assessment on risk and getting ahead of the curve is the name of the game for this century. So the third component of my talk would be very simple to say, we are going to a totally different era. Okay, we are going to basically into a situation that had absolutely no precedent, because so far medicine has dealt only with one thing, is what you have. You build and you manipulate of what you have. So in the history of drug making of human mankind, there was an empirical period. We experienced, we tasted, we experimented, and we moved all along to a scientific area that is very young, only 2 centuries. And on the past 50 years, we laid out foundation, what's called the synthetic biology. It's not anymore just about what your makeup is and how we can work with what you bring into your life. It is what we can engineer and make it different. As we said, the umbilical cord cells from a baby will lay foundation to build your new organs. For example, if you have diabetes and you have no insulin, and sometimes it's type 1, very young people would have it, if you are taking the umbilical cord stem cell, okay, and the person develops diabetes type 1 in the age of 14 or 16, you build his new pancreas. And so also replacement cells for heart and brain. All those cells can be differentiated and be replacement part. So we are going to build new organs and new capability, and in fact you can extend it to say that synthetic biology in the new era that just started, probably in that decade, okay, is going to totally change the prospect of life, because you can choose to actually deliberately say, I want to have better muscles. I have muscles with more capability because I want to win a marathon. So you can extend the medicine into many different directions that are fulfilling wishes that are not necessarily disease-oriented. So going to the next slide is basically summary what synthetic biology can bring about. Many different changes, and the bottom line is it's a quality of extended life. That's what's in stake, because quality of life as it is right now is rapidly declining in old age. And how to preserve extended life with the qualities necessary, synthetic biology can bring with that solution. So the last slide would say another important concept is everybody will have his own disease. If you like it or not, it doesn't matter if you are called asthma or diabetes or Alzheimer or osteoarthritis. Everybody has his own, in a way, his own disease. And therefore, being able to deal with drugs that work for you and drugs that works for you based on your own capability in your own body, your own cells engineered, will be the name of the game. So I think that you need to look forward to this century as being able to actually deliver many of those needs and capability. Thank you very much.
Thank you very much, Dr. Feuerstein. So we've heard in a glimpse already about new technologies, new cells, new engineering methods. That implies that medicine is not anymore an issue of one caretaker and one patient. It requires system engineering. And our next speaker is Dr. Uma Raman Kotagal, who is an expert exactly in this field. The Senior Vice President for Safety, Quality, and Transformation at the Cincinnati Children's Hospital Medical Center, and a very respected scientist in the field of neonate medicine and system engineering on a hospital basis. Thank you for coming
Thank
today.
you very much for the opportunity to present to you. You've heard about the future and it sounds spectacular. But I was thinking as Dr. Argus was talking about that young person who now has a block in his artery who has to go to a hospital to get the care. And what are the possibilities that he would emerge from that hospital with the care that he needed, with the right organ fixed, with no problems, getting the right medications, not getting infected. How many of you have been bungee jumping recently? How many of you have either been admitted to a hospital or had somebody that you loved admitted to a hospital? Well, did you know that the risks of being hospitalized are about the same as bungee jumping? Did you sign a contract when you went in that the risk of you getting infected or being harmed would be significant? Did you know that it takes 17 years from the time that one of my colleagues discovers a new cure for half the people that deserve that cure to get it? So we're sort of stuck here in the present, which is unfortunately not as spectacular as the future looks, but we have to get here from there. And what I want to talk to you about is how we do that. The Institute of Medicine described these characteristics of how healthcare should perform, but in fact, healthcare is unsafe. Half the times we get the right treatment. Sometimes we get harmed. Care is almost entirely directed by the physician or the nurse, not by the patient or the family. And we're spent a lot of time sitting in rooms waiting. Our waiting rooms are bigger than our rooms that we're cared for. So the dimensions of healthcare are really to address this, and these are those dimensions. But how do we get there from here? And to do that, we have to think about some different sciences that are not biologic sciences. They're social sciences. They look at how we will generate new knowledge from basic knowledge to its application. And we do that through creating communities of care. I want to talk to you about a model that we're testing, funded by the National Institutes of Health, where a team of pediatricians and computer scientists are developing a social network that brings doctors and patients and researchers together as collaborators to create this new knowledge. And here we create what we call a learning system. Can a healthcare system be a learning system? Can it learn fast enough in order to apply what you learn today, tomorrow, not wait for 17 years? Can it drive discovery as a natural growth of the patient's views and knowledge, and how can it build innovation? What if it were as easy to find how to create a registry and a practice of find a cure as it is to find the book that you want to order? What if you were able to Google the answer and learn from all of the people that have similar diseases? We could create vastly better systems if we were to harness not only the knowledge of science, but the knowledge of people that experience that science. Well, we were able to collect passive patient-reported outcomes by measuring distances that kids travel with inflammatory bowel disease, and discovered that even before they get their symptoms, they're not feeling so good, they're not traveling a lot. We could begin to think about when we apply innovations in that kind of way. This is a collaboration with our hospital, with the Institute, and with the MIT Ginger.io to create that. What if we built networks of people that got together to learn? This is now about 10,000 children with inflammatory bowel disease that are entering their data and looking at a variety of experiments that they are running, creating common conditions that allow us to learn from each other, contributing to the design and to the technology. What we've begun to do is to identify, using ethnography, what users' goals are. These are persons that describe how they feel when they have a disease, what they would like to see their functions look like. And this for example, is Bianca. Bianca is 20. This is how she is. This is who she is. This is how she approaches her disease. And her goal is to keep the symptoms at bay. So Bianca might think about a way in which she could be a leader in this community by connecting mobile applications, using games as social engagements, by mentoring programs where people could mentor each other, by keeping local community building efforts, and by applying the science that you heard from my colleagues in this way. So this is a goal of you, me, IBD that connects people together so that they could be running their own experiments, and we have a number of applications being developed in this regard. This is one example of an experiment. This is an N of 1 prototype. This is a single patient collecting their data, recording their symptoms, keeping track of their journals that allows them to test and understand what medications or things they use make a difference. This is what a system would look like that continued to learn, that had the data that could be logically analyzed, where patients entered their own data, but where we pooled a lot of information and where experiments happened real time. So that's the system that I think will be the system that we now are creating, that would have worldwide communities that would learn together about a disease, that would have applications for individuals for those diseases, and that where new knowledge and new drugs, such as you heard from my colleagues, could be applied to continuously learn. Such a system would be a long way from the system I described earlier, where it takes 17 years for half the people to get the right knowledge without getting harmed. I'll take questions at the end. Thank you.
Thank you, Dr. Kotagal, and we will have an open session for questions after the next talk. And the next talk is presenting the way we speed up discoveries, which is an essential requirement to reach the processes that we were discussing. Our speaker is Dr. William Linton, Bill, who left a career in research to develop research tools for the benefit of the scientific community. The chairman and CEO of Promega Corporation in the United States, the president of the Biopharmaceutical Technology Center Institute, director of the Wisconsin Technology Council, and of Analytical and Life Science System Association. Bill, thank you for coming here.
Thank you, Mona. Shalom and good afternoon. I did found Promega about 34 years ago. My training is in biology and chemistry. Professionally, I do manage this business, but I have a very strong personal interest in ideas and research that explores the frontiers of science regarding the mind, brain, and notions of reality and world that is within us and also outside. And I want to talk about how this perspective can perhaps inform us a bit on the future of medicine. And thanks very much again, Mona, for this opportunity to share a few of these ideas. Our business in the past 34 years has been focused on molecular tools used in studying and bringing better mechanisms and methods to the scientific community, which obviously has implications in healthcare and in human diagnosis. I want to take just the remaining time to create a perspective that's perhaps a bit different and gets us a bit out there, but I'll bring it back in just a moment. So a very long time ago, in fact, it was about 14 billion years ago, there was really nothing. There was nothing there. And then there was a quantum fluctuation in the midst of no time and no space. And then, about 400,000 years later, the universe very rapidly inflated, resulting in this thing that we call the universe. And at first there was just hydrogen and helium, and these two elements condensed into billions of large local balls of superdense matter, in which the fusion reactions occurred and stars were born. And elements only up to iron were made in these reactions. And then 5 to 6 billion years ago, one of the stars began running out of fuel. It expanded to a red giant and then collapsed on itself and exploded in a supernova. And in the supernova, like billions that have occurred elsewhere in our universe, all the other elements were created. And from the death of these stars, the elements of life were created. So planets were born. Life appeared on our planet about 3.5 billion years ago, becoming more diverse, more organized. And eventually, neuronal sensors tapped into the environment, enabling growth, reproduction, energy uptake. These neurons continue to multiply and organize, culminating in a remarkable organ that sits on top of our skeletal frame, directing, deciding, and contemplating. So 100 billion neurons, and altogether about 100 trillion neuronal connections working in exquisite coordination, ultimately turning inward to ask the question, who am I, what am I doing here, and by the way, what's going on out there? So the universe has become, in a way, self-aware through itself and through each of us that are here today. And I'm going to come back to this point in just a moment. So the question that's being asked today is predicting how healthcare and life sciences that we study can support medical research. And these predictions are pretty tricky. And as Kahneman said in his talk earlier this morning, the forecasts are often mistaken because of one hidden assumption, and that assumption is that tomorrow will be more or less similar than what we have today. So we tend to kind of build on what we know and then extrapolate that into the future. That's about the best that we can do. And of course, the problem with that is that the knowledge that we build on is expanding exponentially. All of the information, all the technology is increasing at an ever more rapid rate. So it gets more and more difficult to actually predict what's going to be happening into the future. We might consider that the beginning of the age of molecular medicine kind of came about when we understood the basic structures within the cells, DNA molecules, discovery of codons, roles of genes, proteins, and the intricacies of cell signaling. And of course, we look at that and we take a lot of pride, in what we've learned through that. But I just want to remind us that we occasionally have to step back and look at this with a certain amount of humility. And a Nature magazine article that was published just last month, a number of scientists, or about 300 scientists from, I think, about 14 countries, published work on their outcome of the work regarding the chromosomes in tomatoes. 35,000 genes in a tomato. Well, let's go back to 1969. And in 1969, it was thought that there were about 2 million genes in the human. By about 1990, it was thought, well, based on research that they had done, this actually came down to about 125,000 genes. And then just a few years ago, that number came down even further, surprisingly, to about 25,000 genes. So I think we have to have a lot more respect for this tomato. 35,000 genes in the tomato, 25,000 genes in the human. And that would have been very surprising had we suddenly forecasted that 20, 30 years ago. Well, we know today that there are a lot of reasons for why that happens. And as we study and understand more and more of the mechanisms, as was mentioned earlier about these microRNAs, we also understand in humans and in higher life forms, there are a large number of intricacies that we just couldn't even imagine just 20, 30 years ago. And again, this is how quickly things are changing, how quickly our perspectives are changing. So what are the implications for the future of medicine? And again, going back to my original slide that shows the human brain. Keep in mind that in the human, a very large number of genes are directly associated with the nervous system. So regarding today's topic, I'd like to just mention one factor that can have an enormous impact on our health, and that is stress. Stress is a fact of nature. These are forces inside of us, outside of us, that affect ourselves, individuals. And the way that we respond to stress can have an enormous impact on our health. Because of the overabundance of stress in our modern lives, we usually think of stress as being negative, but in fact, from a biological point of view, it can be negative, neutral, or positive. Stress is just one factor among many that are induced and managed in some fashion by the human brain, by our state of consciousness, and how that impacts our health. So if you just think about all the number of things that can be impacted on our health by this idea of stress: headache, muscle tension, pain, chest pain, fatigue, stomach upset, sleep problems, depression, a whole variety of things. You can list pages of things that can result from stress. And if you think about it, stress itself is something that we create from how we respond to our environment. We treat many of the downstream symptoms, but I'd like us to consider our state of mind and how we respond to things and how that can dramatically impact on our state of health. So managing stress then, by changing our perspectives and our outlook, in effect, by changing our minds can have a direct and lasting impact on the body's response, minimizing the negative impact of stress in many ways. In summary, changing the state of mind directly affects our health and can affect our treatment options. I think the next several decades will give us profound discoveries that will continue to link our well-being and our state of mind. So in the same way that quantum physicists were really astounded and somewhat dismayed to learn that a conscious observer influences the outcomes of what they thought were purely objective experiments, I think we too will discover profound power within human thought, mindfulness, and consciousness on every aspect of our health. Thank you.
Thank you, Bill. That concludes our opening presentations. And I would like just to add one comment. We are all talking about new developments, new engineering, new systems. We are not sure yet that all of those are fully justified. And there's this story about the guy who got one of these new electrical cars, right? This is Israel. We look into the future. And he's driving from Tel Aviv to Haifa, and suddenly he sees a chicken with 3 legs that runs faster than his new car can drive. And he's getting really upset. So he drives faster and faster and faster, and finally the chicken gets off the highway into the back road and disappears behind a barn. And the worker comes out, you know, jeans, checkered shirt, and the driver says, "Excuse me, I thought I saw a 3-legged chicken getting into your barn." And the guy says, "Oh yeah, we engineer them." The driver says, "Why on earth would anyone like to engineer a 3-legged chicken?" And the farmer says, "Look, there is me and my wife and my son. I like a chicken leg for dinner. And so do my wife and son." So the driver says, "Are they tasty?" The farmer says, "I don't know. We never managed to catch one." So that concludes the serious part of our presentation. And I'd like to open the panel for questions. Please say to who you direct your question. Please.
Hi. My name is Rafal Golodensky. I'm from Hadassah University Medical Center. And I want to comment about Dr. Holstein's lecture, of course, about the future medicine, synthetic medicine, stem cells, and so forth. I think we have stem cells already in 2 decades, 3 decades. People are working on stem cells. We have all kinds of stem cells. I don't want to bore everybody by describing them. Still, not even one tissue engineer is really, a tissue is really functioning. So the question, is it really a hope or a hype? That's what I myself have to admit I work a little bit on stem cells. But there are studies that show the stem cells do have some effect. If you inject them, eventually people feel better, different diseases. I don't want to get into details. But the effects is only as a bystander effect and not really by engineering the tissues. Then comes the question, should they be stem cells? Because stem cells means that they can differentiate to make all the tissues, and they just secrete something which is like a pharmacy that you inject into the patients. So I just want to put it in the focus that some of our expectations that we take it for granted. Yes, in 2, 3, 5, 10 years we'll have new pancreas. Well, it's not straightforward, and so far it hasn't justified itself, and that's what I referred to Professor Sorek's remarks here. Thank you.
So that's not really a question, I guess it's a statement. Okay, so, but the good thing about the question or the statement is you mentioned the word hope, right? So the century just started. I think the fundamental technology is available in prototypic way, and if the thought is correct that you can engineer and you can use your own body to plan ahead years to supplement decaying cell or organ and then replace what you need, all the science that will allow that to happen is in its fundamental component available. The century would have to put them together and learn what is working and what is not. Like everything else, it's a reiterative process. It's a market-driven process. It depends on the successes. But everything is possible in that direction. There is no reason why you will not use your own cells that you have been born with in order to produce a new organ that you don't have anymore. And living without a pancreas, for example, is not convenient. That kind of thing.
Question is for Dr. Agus. Are we likely to see a revolution in our understanding and treatment of psychiatric disorders such as ADD and ADHD?
It's a great question. So by the way we're doing it now, no freaking way. We try to be reductionist, right? Let's figure out the one gene for ADD or for autism, et cetera, and realize that we are a complex emergent system. Looking at one dimensionality, which is DNA, which is your ingredient list, is not going to tell you what's happening with these complex diseases. So we have to take a step back and look at that system. We have to take a step back with these newer technologies and look at the dimensionality. Take cancer as an analogous example. They say, well, molecular targeted therapy is going to be the future for cancer. And again, I'm a cancer doc, and that's gobbledygook. Unfortunately, the average cancer on diagnosis has several thousand mutations. You can't model it, you can't target all of them. And so this notion of this reductionism to one element is probably wrong. We have to start to think about it, again, more like a physicist and engineer as this complex system, and then I think we can make real progress.
Okay, I'm Danny Fulton from Israel. I'm dealing with health economics. I would like to ask all your beautiful presentations how the world can support all this while we are hearing all the economical crisis, all the what called the old economy, the E7 countries are weakened, all the economy is going to the the G7, all the economic are becoming weaker. The economy is going to the other side. Maybe China will be the leader. And how does it go with not enough regulation in those countries, specific to what Raman told, and before Giora, if we wish that more governmental aid will be in. Thank you.
I think it's a great question. Certainly in the U.S., the economy and the GDP, a percent of GDP that healthcare consumes is untenable. It's largely untenable because we're still enamored of savior approaches. You know, you're really sick and I could rescue you, versus fundamental, very boring, very simple beginnings to improve the area of population health. And we are starting to see that cost curve bending, actually, as people begin to make better choices. We are certainly looking at experiments that link healthcare with social strategies, much more than thinking about healthcare as an isolated, sort of great therapeutic solution for the world. So much of the conversations around this, in our case, involves combining social agencies with healthcare agencies to address some of those modifiable factors that I think people have referred to. So, for example, I think we know that that for asthma in children, which is the commonest chronic illness, that we find many of our children come back to the hospital not because they don't know they have asthma, but because they live in families where they may be living at the weekend in someone else's house and they don't have their medication, or they actually don't get the medicine, they don't pick up their medicine in time. We're able to look at those kinds of therapies and go farther upstream to solve those problems. So I think we're seeing a lot of emerging models, you know, in that arena. Certainly seeing that in a lot of the North European countries whose outcomes are considerably better at much lower cost. And the classic example I'll give you is in Sweden, where they have a kind of a persona called Esther. And Esther is an 80-year-old woman. She lives by herself. Her daughter lives, 2 cities away. When they discovered all of the hospitalizations for hip fractures in this county in Sweden, which has the lowest cost and the best outcome, they discovered 2 primary reasons for hip fractures. One was that the light bulb was fused, and the people were standing on the ladder to fix the light bulb, or the light bulb was fused and they decided they couldn't stand on a ladder, and so they were going to walk down the stairs. So in this county in Sweden, they change the light bulbs on a regular basis. They've got a young somebody that goes and does that for not a lot of money, and they've nearly eliminated fractures. So I think while we simultaneously pursue these great, important opportunities of science and new discovery, we must get back to the conditions and issues of preventable illness that are largely addressed not through healthcare but through a broader commitment.
So actually, I'd like to, raise a question on this. And a very large percent of healthcare costs are often consumed in the last year of a person's life. It's enormous because there seems to be such a huge focus on the quantity of time, and we alluded to the fact that people are living longer today, but it seems to me also that the quality of a person's life is an extremely important factor. And is there a relationship, between this focus that we have on extending life and the cost of healthcare versus the quality of life, and how that might affect ultimately how much we spend? There's a question.
I mean, look at it this way, is that there's this magic number of 83. Once you pass 83 years old, we don't put you in an intensive care unit. We don't go to heroic measures. We actually let you die with dignity. Remember, 1953 was the last year in the United States you could die with old age on a death certificate. So if we prevent disease and make people live into the 80s or 90s, the paradox is healthcare costs will come down dramatically.
Hi, thank you so much. My name's Aviva. I have a background in Western medicine, alternative medicine. I currently work for a biotech, biopharma company. I just really want to say thank you to all of you. It's so comforting seeing brilliant people on the inside looking to make changes, because there are so many of us that don't know that that's happening and have that fear. This is pulling from everyone's presentation, but Dr. Kodagal specifically, on seeing what you see within the hospital system. Those of us, I don't want to say at the Mercy, because I do think that there's a big push now in alternative medicine as a result of seeing that medicine is not reaching the needs of especially the people with chronic diseases. And there's a bit of intuitive medicine that's really coming out of this. How do you recommend that those of us who need to go into the hospital put our trust there? Really, like I had a family member go in recently with a stroke, and the only thing that kept me quiet was knowing that God is the chief medical officer of that hospital, and speaking to everyone from that perspective. Otherwise, I would be in a complete panic. And I know personally, I only go to my doctor when I know the exact drug that I want him to recommend to me, and everything else I take care of myself and do a better job. And he's a brilliant man, and he's a wonderful man. I have his personal cell phone number and call him in the middle of the night sometimes. But I don't really know what to do with that, or how to raise my child with that, or what to do in my community with that, and I would really love to hear what you have to say.
I don't want to be the depressing person in the room, because I feel pretty optimistic most of the time. But I would say that I would not trust your hospital. And that's a fundamental principle that you should follow. Not because that physicians are bad or nurses are bad, that our systems are perfectly designed to get the results that we get. And unless we fundamentally change our systems, it will be that way. One area that we've been exploring where I think there is knowledge around how to do this is really the nuclear industry, the aircraft carriers, and so on. Where application of reliability engineering and reliability science makes a difference. These are called high reliability organizations, and they have been extensively studied, and they have some fundamental concepts that define how they work. And the most important part of it is, I think somebody referred to earlier, is these are complex adaptive systems, so they should be recognized as such. But using the high reliability engineering science, we have been able to make pretty dramatic impact on modifying human systems to make them more significant and more reliable. And a lot of that really depends upon prediction as a criteria. We do find that we are able to predict, if we apply the right theories, where somebody might get harmed, and we are able to mitigate it. And there's a whole science emerging around that. Healthcare, like firefighting, though, is a very unforgiving environment in which experimentation is difficult. And so under those circumstances, how do you experiment to create a safer system? And there's an emerging literature on that is quite interesting and promising. In the meantime, I would recommend you have somebody sit by your bedside if you are in a hospital. I would recommend that somebody check the medications, and I would recommend that you follow fundamental safety principles that you would follow in your home.
Shaishin Orr, I'm a faculty member at Tech, and I'm a systems biologist working essentially on the immune system and the complexity of it. And I guess the questions to the panel, on one hand, I fully agree that humans are complex, and yet the systems in which we design our drug pipelines these days require large numbers. So if everything's personal, how can we, in the end, have the entire pipeline system be, averaging numbers, or in fact, how would this be cost effective? Would even the current regulation system of the FDA be relevant if the treatment is personalized?
Yeah. I can take a stab at it and to say that that's a very common concern about fragmentation of healthcare to the individual. I think that's taking it to a great extreme, but the reality is that in all major diseases that we suffer, 95% of the GDP spent on the chronic human diseases. There is a clear need to define the disease not on the clinical manifestation, like, for example, somebody comes into the hospital and is wheezing, he's called asthma. But the disease on a molecular level, it's not about wheezing. It's about certain biochemical changes. And it's very clear that there are patients who wheeze because mechanism A, patient who wheeze on mechanism B. And if you treat one that has mechanism A for a drug that works on mechanism B, all that you do for the patient is adding side effects for no or limited benefit. So the economics of putting compounds into people that have no benefit needs to be factored in, and segregating 19th or 18th century terminology of what a disease is based on phenotype rather than based on the mechanism, okay, is going to change. It doesn't matter how objection or what concern will be raised. The economic will come in once you prove that you can have, let's say, a quarter of the patient called asthma being treated so effectively and without no side effect. Those patients will demand that medicine. The physician would treat only with that kind of medicine. So today the treatment with asthma is for all comers and only few benefit, and many get side effects, including cardiac arrhythmias. In the way asthma is now being progressed, and certainly in the next 10, 20 years, there will be a called, my asthma is this kind of an asthma, and for that I will need that disease, and it will work very well for me, and I will be very safe. So I think it's premature to benchmark the economics of that situation, but the demand by patients and doctors will dictate it anyway, and the industry can kick and scream about it for concern of we cannot do medicine for everybody. That's not going to happen.
Yeah, I'd like to add to that. Many years ago, I used to go to the pediatrician expert with a sick child, and he would write something and say, okay, give this to him 3 times a day. And I would say, could you please let me know what it is? And he would say, it's a good medication, it's imported. So I would say, could you please let me know which medication it is? And only then he would raise his eyes from his papers and say, oh, it's you again. I think that that situation has changed. Today's patients know a lot more. They can Google, they can look into the literature, everything is accessible, and they come to the doctor and say, I've read this article recently, and I think that I need this. And the doctors listen. So there is a major change here as well. And talking about numbers, I have a friend in University of Chicago who says, you don't start a genetic experiment until you gathered 10,000 volunteers. So that's not for a small country, I'm afraid. Yeah.
Anna Szabolcsi, I'm a medical doctor, pain specialist in London. The question is to Professor Agus. We mentioned about the new research on identifying the microbes now, which are modifying hormone metabolic process. What type of illnesses you can predict from the identifying this microbe, like diabetes or metabolic or hormonal illnesses? And the other related to this, you mentioned in your previous lecture that aspirin and statins are cheap, and it's important to give it widely to prevent illnesses. The same way as it's so expensive, or it's early stages to identify the microbe, would you suggest probiotics the same way?
It's a great question. I mean, remember, with any disease, all the common diseases, there's a genetic component that could be anywhere from 10 to 50%, and there's the environmental component, of which microbiomics or the bacteria are part of it. And so there's that interplay between all of them. Microbiomics is a totally new field. Literally in the last month and a half, there was 5 papers in Nature, Science, and one other journal showing that we can start to characterize them. So your answer is, you know, what percent of what disease are correlated? We don't know that yet. We're going to know that over the next 12 to 14 months. The studies are ongoing, and their guarantee will be a component. So you'll have a genetic component, a microbiomic component. There will be multiple different dimensionalities. And should you take probiotics? The problem is I don't know what probiotics are. Right? I don't know which bacteria they are. And even if they say it's a particular bacteria, what we've learned is there are hundreds of subtypes of each bacteria. And so the complexity of probiotics is such that we don't really understand them yet. We know in some cases they work, right? If you go on antibiotics and you take probiotics, you do much better from GI symptoms and from resistant bacteria. It works clearly in that setting. In terms of probiotic to prevent common diseases, heart disease, Alzheimer's, and others, we just don't know how to manipulate the specific probiotic yet. But I guarantee you that day is going to come.
Thank you. My name's Caroline. Question for Dr. Agus and Professor Feuerstein. Where do you see medical ethics and perhaps the human psychology coming in as obstacles to this positivist approach where we pre-screen for everything and can prevent illness? From your example of having a real live heart disease screening and from your examples of preventing diseases before the embryo's out of the womb. Thank you.
So it's a fantastic question. I mean, to me, medical ethics is something we need to bring out of the closet and talk about. It's very easy to rub these things under the carpet. You know, why did stem cells take off? Because our first President Bush, second President Bush was awesome, and he said, I'm going to ban stem cells. Because of that, everybody in developed countries knows what stem cells are. We all talk about them. There was a lot of hope to them, and it's all because Bush tried to outlaw them. He brought the conversation out of the closet, out to the public. We need to make those conversations happen. We have to bring them to each of us so we can debate them and together come up with answers.
My take on the ethics issue is that ethics will evolve with society. It does so anyway in every aspect of life. We have different attitudes to segment of population today that have not been there 10 years or 20 years. So ethics is part of what society is about. It has no particular angle to technology because technology and science are totally agnostic to a moral or ethical content. It is society that decides what kind of etiquettes of necessity or unnecessity will be introduced into, for example, synthetic biology. For example, if I have today sitting here as muscle systems that does not allow me to win a marathon championship, okay, I now want to have synthetic biology do the job for me and equip me to fulfill my ambition in that regard. Is it ethical? Are physicians going to fulfill that kind of a wish? Well, there are already drugs on the market and procedures on the market that physicians modify people's ambitions anyway, and society seems to accept it. I think the ethic issue is not an ethic of science and technology because they have nothing to do with it. It's what society will do to their interest in health and progress in any particular area as it is in other parts of life.
Yeah, but it's also education, right? If I gave you growth hormone to a 70-year-old, they're going to look better, they're going to feel better, their friends are going to go up and say, you look awesome. And so that 70-year-old says, I want to look better, I'm going to inject myself with growth hormone. There were $2 billion sold in the United States doing that last year, by the way. Those people will live on average 12 years shorter. So again, it's what's your metric? And it's what are you following long term? And education's a big component.
I mean, I was thinking about the barriers of a woman getting breast screening for the gene of breast cancer. She might be able to know, but she might not want to know. And how does that not in your way of presenting?
You look at the data, and study after study, people who know have a better outcome. So that's been shown with Alzheimer's, and that's been shown with breast cancer. They actually feel better knowing. Every woman with a genetic risk of breast cancer has a family member who's had breast cancer. So in the back of their mind, they're worried about it. When you start to do the test, they're empowered. If they have it, they can do something about it. They can work for a cure. They can raise money. They can have prophylactic medications. If they don't have it, they feel relieved. But in both cases, I think they do better.
Hi. Concern of ethics is important because you say, if somebody knows that you have a predisposition and risk, he can decide that you are not employable, can decide that you are not fit to a certain job.
Well, but that's not true. I mean, there's a Genetic Non-Discrimination Act that was passed by President Bush, one of the few things, again, he did that was very positive. And so that's outlawed now in the United States. There is yet to be a case in the world based on that.
Hi, my name is Ariel Beery. I'm in the business of training social entrepreneurs. And I was very fascinated by Professor Kotegal, by your work, and also, but in general, the data component that exists. And you're all here in the state of Israel, which is renowned for its ability to create startups. Particularly those startups are not the deep science and research that all of you are dealing with, but things that take existing data sets and then create apps for them. I'm wondering maybe you can give some of the people in this audience that I'm sure are involved in the startup economy a few tips or insights or thoughts as to what are problems that Israeli entrepreneurs could be working on to bring the future of medicine.
I could start with a couple on my mind. In the acute care setting, there is a need for us to recognize, predict, and mitigate deterioration so we could pick it up at a much earlier stage. If we look at harm, at least in hospitals across the United States, and I believe that it would look very similar here, in average, about every 17 days, somebody is harmed seriously in a hospital. And by seriously, I mean they either suffer death or significant harm. Of those, about 60% are related to deteriorating conditions that are not picked up. So a very important piece of information is really figuring out how we recognize, through using the EMR, some of those situations. I think another important one is the one I referred to around applications for passive patient-reported outcomes. How can we collectively harness the knowledge that allow us to look at precursors and pre-disease states? I just stopped with two of them because I think there are lots of other examples.
I'm just going to add a comment. If I were an Israeli entrepreneur and I heard and had looked at the study that just came out on the human microbiome, I'd start to track that and follow that because that's going to be an area where I think there are enormous business opportunities because very few companies in the world are at all focused on that. I think that's going to be a large emerging area.
And I would like to add on that there is a change in the pace of progress. That is, in many cases, science today develops much faster than the set of clinical trials for developing a drug. And that inconsistency is one of the reasons for the very slow pace of development of finally approved medications. So things like the model that Dr. Feuerstein was mentioning are a necessity, an essential necessity to speed up development and follow the pace of science. Yeah.
Okay, first of all, I have one remark regarding what Professor Sorek said about the doctor that wrote the medications. Now doctors can't, and this about the conference as well, because everything is about tomorrow and computers and everything. A doctor can't scribe something on a piece of paper that nobody can read, that what used to be. Now it has to be printed in a computer so everybody can know what they're taking. What I was talking about is everybody's talking about how beautiful our species are and how big progress we're making to tomorrow and Facebook and computers and technology and everything. But there is no mouse with Alzheimer. We have to create it to make a model for our research, but there is no mouse, there is no rabbit with Parkinson's. Why can't we just learn from the past, from evolution, how the animals and what happened from there to now? Many of the genes were improved by evolution and by selection and by everything, but there were probably a few that were harmed. And there was a price to pay, and I believe that we can actually learn from mechanisms and family of genes and systems biology of other organisms to know what is wrong with us, that we get cancer, we get IBD, and everything else.
Well, I absolutely agree. It's a great issue. And we get sick because we would like to live longer than those animals do. The Homo sapiens is the only species that outlives the reproductive years. So rabbits in the wild would never survive to an old age. No wonder they're not sick. You know, in the 50s, Japanese used to tell that they don't have neurodegenerative disease, and they didn't. They all died of cancer of the digestive system because they were polishing their rice on asbestos. As soon as that was abolished, life expectancy in Japan jumped in about 30 years. And today, in hotels in Japan, you find diapers for adults because they do have an aging population. They do have neurodegenerative disease. We are studying those diseases, and there's a lot of effort being spent. As I said, I think that much of the effort should go back to developing early diagnosis, and that would be the opening for newer and better therapies.
My name is Aaron Goldenberg from Florida, and I'm on the MSAP program Career Israel. I've read a couple articles on research that's saying that things like a walnut that resembles a brain, or the tomato, when you cut it open, it looks like the chambers of the heart, or the carrot looks like the cornea. And when you consume those things, it seems to help with brain function or your heart. Are you guys doing any kind of research taking clues from nature and applying it to your own projects?
I can take that briefly to say a couple of people are concerned between the congruency of the human disease and biology and modelings that are being used to study human disease, and that's clearly a gap and a concern. Every scientist in biology has that issue to deal with. So there are, as has been said, ethical limitation of what you can do either on humans and on animals too. And that clearly slows down and impedes the ability to get a more precise solution or insights on what human disease and biology are about. But for your specific question, we learn about the way neurons, for example, die. Originally, the phenomenon of apoptosis was discovered in an earthworm. and that laid out really a very intense research on what's called programmed cell death. And there are certain earthworms where you can know exactly the date since birth and the time in the day where a certain neuron will die, because it's programmed within that animal. And that phenomenon of how cell can die by their own internal mechanism inside the cells, and how activation of those mechanisms define the fate of the cell, are well known, and all that knowledge has come from a very primitive so-called organism, and drugs that are intervening with that process are being actually produced. So it's a good question, and how to translate into the human situation and test it and then produce long-term clinical prospective studies is a huge challenge, again, primarily because of economy reasons.
Hi, my name is Ofer Fainaro from Hillel Yafe Medical Center and the Technion. And my question is for Dr. Agus. With regard to cancer prevention and screening, what do you take into these new dogmas that are rising that we're probably over-screening for patients? Let's say mammographies are, the age for mammography is probably going to go up. We're detecting too many early diseases, and we don't really know what would happen if we would not treat them. Let's say if we take biopsies of thyroid glands from people who died from any other causes or motor vehicle accidents, we can see that 90% of these thyroids have small tumors. So are we focusing too much on early detection, but rather than moving on to managing cancer, making it a chronic disease, the ideas of Dr. Folkman, the late Dr. Folkman?
It's a brilliant question. And so there was a study done in New England Journal of Medicine where they took 6,000 men and they said, get a PSA test every year, get an exam every year, and 6,000 said, do whatever you want. At the end of 6 years, there was no statistical survival advantage, and therefore the US government said, throw out screening, we don't need to do it. What they didn't realize is that 74% of people in do whatever you want arm got tested every year. Certainly, we overtreat cancer. The other side is countries that screen for prostate cancer, for example, have half the death rate from prostate cancer at countries that don't. Since we started screening in the United States, death rate is down 47%. 35% of prostate cancers don't need to be treated. Clearly, we know the biology. Same is true for thyroid cancer and some breast cancers. So the argument shouldn't be throw out the baby with the bathwater and stop screening. The argument should be let's use art, which is hard because it's not binary, black and white, and we can diagnose cancer that we don't need to treat. I've got 1,100 patients in our clinic with prostate cancer we haven't treated that we're following actively. We call active surveillance. Right now, Korea, the highest incidence of thyroid cancer in the world. Why? Because it's the highest incidence of plastic surgery in the world, and they examine the thyroid all the time, and they find lots of cancers. Do they need to treat most of them? As you alluded to, no. But the art is knowing when to treat.
I'd like to address that point maybe from a very different angle, not necessarily contrarian, but from a scientific point to say that everybody who walked to this hall looked in a certain way, and when you walk out of the hall, you will think that you are the same. But genomically speaking and metabolically speaking, everybody in this room is already different since he came into the room. The point to be made that our blueprint, our DNA, changes sparsely. Have a mutation, we have somatic mutation, germ cell, maybe every decade or some years. Other things change on a scale of hours and days, and the metabolomic will change on a scale of second and sub-second. So, if we know that we are not the same constantly, and we can go back and forth on a baseline in a different format, and we don't think that we are changing, the very essence of our format of life, the way we live is that everything changes all the time. And if you want to detect your disease or you risk for the disease, that train of thought forces you to be for your personal sake, not from population economics or ethics or convenience. From your personal situation, you want to be checked as soon as you can, all the time. Because you do not know when you will get a certain mutation. You will not know when your proneness for cancer has incurred an additional critical event that will make you now a cancer patient. So from the ideology and the makeup of our life is that if you would have a system that will check you, for example, for 1 penny a day, every day you will go to be checked for your total being in order to make sure that you will catch a detrimental event as soon as possible. It's not practical. It's not even imaginable, but the fundamental of the biology of human and animal is that you change all the time, and it's in your best interest to detect your fatal change possibly as early as possible and manage it as effectively as possible.
I think it's not an ethical question, because the real question is, are we doing good with this screening? Dr. Agus mentioned the PSA. Michael Karen published in Nature that if you biopsy the prostate, you actually worsen the disease because you get an influx of hematopoietic cells. So you think it's not true?
Not true. That has been proven wrong.
I think that we will conclude with conclusion that not everything that we read is true. And that we are all changed. Thank you for being here.